Well, hello and welcome. My name is Jessica Castner. I'm the president and principal of Castner Incorporated and the project director of this workshop. In this segment, we're going to talk about the NIEHS Translational Research Framework, or TRF. I want to acknowledge the funding of NIEHS to support this work. So here's an overview of the organizational of this lecture. We're going to talk about why do we do this? Why are we learning this? We're going to talk about the details of the TRF. We're going to talk about applying those details to your research trajectory figure, give examples of other neuroscientist career cartography, and talk about preparation for how we're going to use time in the workshop for you to create your own career cartography from the TRF. So in this pre-recorded lecture, we're going to spend about 20 minutes working to understand the translational research framework, and then we're going to use it in the research framework to create a community of practice and dialogue about what it means to you and how you can create your own cartography, which is a figure or a map of your career, your research program or trajectory. So let's talk about the TRF as career cartography. This is a useful tool to accelerate career communication and trajectory that integrates exposure science. It helps to think about the next steps in planning for purposeful collaboration, what other labs, skills, or tools you need to move your research forward. It's helpful in reflective evaluation as well as planning the next step in moving forward, and it gives a clear visualization of what you've done and where you're going for both your own reflection but to communicate your scholarship to others. So before we start, step 0 of creating a career cartography from the TRF is identify and write down your own research trajectory mission or that project statement. Often we do this when we're putting together a biosketch, our web page, our CV. My example I give here is the multi-level measurement and reduction of airborne pollutants with emergency effects on respiratory and sense organs. The process we'll go through here is published, and that reading is uploaded in the virtual classroom here in the Nursing Outlook manuscript that came out in 2020. So what are the materials and methods needed to create your own cartography from the TRF? Your CV, your program portfolio, consider your grants, your pubs, your presentations, policies you may have contributed or written to, program descriptions, those outputs that you've created related to an environmental health program of research. What are the methods? We're going to talk about the 4-step methods in this talk. First is to identify translational categories and activities. These are rings and nodes. We're going to talk about that in detail next and link that to your program of research deliverables or your education deliverables. Within each ring, you're going to link specific works and program outcomes to these nodes. Again, we'll talk about that in a minute. Then you coherently depict it as an overall map or figure. We'll see some examples and we'll want you to create your own in the workshop. And then last, develop and recommend improvements back to the developers of the TRF for program cartography utility or potential improvement of this shared mental model. Here are the 5 main categories of the TRF. And you'll find in our virtual classroom, there's a guest YouTube where Christina Pettibone, who was the lead author on generating the NIEHS-TRF, explains that process to us. So optional content, but I just want to thank Dr. Pettibone for her engagement with us as environmental health nurses. So what are the 5 main categories? Number 1, fundamental questions. Application and synthesis. 3, implementation and adjustment. 4, practice, and 5, impact. We'll go through examples of each of these in detail in the following slides. We tend to understand as nurses or as clinician scientists the translational continuum in the health professions as following what's usually medication or device development in clinical trials. You've got phase 0, Preclinical research, basic research. Phase 1 is your clinical safety, dose, feasibility, and measuring potential adverse effects. 2 and 3, where you're doing your clinical trials, and 4 are those post-market clinical trials, all the way to the implementation of standard practice in population health. The NIEHS-TRF can be useful for these types of clinical testing trajectories as well, but it's expanded to include environmental exposure research and considerations, and really puts a unique lens on that gap for those who might struggle to look at how environmental exposure and that interdisciplinary component enters into health and clinical research. It folds population health observation, where that tends to be T4 on the clinical research translational. It folds these observations back into fundamental questions in a really useful way that's coherent with how many nurses utilize epidemiology and secondary data analysis of population data sets. So let's start in that center ring. Fundamental questions. This is ascertaining the fundamental mechanisms of biology, disease, or behavior in traditional explanations. This is your basic and bench science. What's special about this ring in the TRF is the shared mental model integrates research questions, experimental settings, and organisms. For nursing science, the other rings will mostly focus on human beings as the organism of study, but environmental exposure science includes a wide range of plant and animal research that can drive human health discovery and insight relative to environmental exposures. Fundamental questions discovery may incorporate human, animal, plant, bacteria, yeast, worm, fish, or all kinds of other model organisms in the discovery phase. A great example is with the NIEHS Superfund podcast. It's in 2020, and I'll go ahead and reference that in the virtual classroom, but it talks about testing edible sorbents to protect against metal toxicity in animal models. So here's a new potential treatment for poisoning. The background is an emergency nurse. I'm very familiar with sorbents and with acute and poison exposures and the need for nursing interventions there. So what about longer term metal exposure? So that potential for it not to be absorbed in the body is exciting on these fundamental questions. Fundamental questions focus on identification, what is it? Observation, what's it doing? And understanding, how does it do that and what else is going on? Settings and fundamental questions may be the laboratory outside the normal bio context, like the test tube, outside a living body, inside a living body, in computer model designs, or in the original context, or an observation among the entire population and group of study. Geospatial mapping is included here as observation, particularly of exposure or of particular health effect. Many nurse scientists will conduct secondary data analysis of population representative data sets like NHANES in their fundamental question work. Again, this is at the population level. We'll address some of these methods in our informatics modules for you. Note that novel sensor or tool development may belong here as well. Bench and laboratory science, again, is focused, and we do know that engineering has a huge component of this work in the interdisciplinary space. Dr. Rodriguez, one of our instructors, does a lot of work in the microbiome and biomarkers of exposures. And so a lot of her work that she'll present deals with this space. I've looked at some analytics innovations or, some new ways to do the mathematical modeling, especially with temporal indicators that would be, a type of modeling that answers some fundamental questions. So after we just introduce everyone, we'll come back to this big picture to contextualize it. So notice the center rings are those 6 research activities or nodes we just discussed. The first step in career cartography is circle the nodes to relate to your own research trajectory or plan. Take and map what project you may have completed or are planning and set it in here. Where does it fit? You may not circle any in the first inner ring, or this may be the entire focus of your work if you're in more of a bench scientist or a secondary data analysis space. This is helpful to also consider Where are the gaps once you finish this? What other labs might you want to connect with? Collaborations, partnerships, interdisciplinary reading, or conferences that might strengthen your own science and pathway. Something in the fundamental questions might be sort of a blind spot in where you've been navigating in your career, and it's helpful for that planning as well. So the next ring is application and synthesis, denoted by these light blue ovals. These are controlled trials and experiments. The setting is structured and predictable, but these are not yet in general hypothesis testing. They're generally exploratory or feasibility testing work. For examples, it may be the pilot tests of an intervention, methods and approaches, new tools. Systematic review and formal synthesis are included here. It's important to know that a great deal of regulatory science at the USA federal agency includes rigorous and comprehensive research synthesis to inform policy. An example of this is the Environmental Protection Agency's Integrated Science Assessment. Some other examples that we can present and send you published literature on, Dr. Huntington-Moskos has done some data collection method testing among adolescents in particular with some new tools to test exposure. Our team is currently working on a Cochrane systematic review looking at disasters as the exposure, so all hazard and the household preparedness. We have also looked at the feasibility of Fitbit as a wearable sensor to get a proxy for sleep quality and sleep disruption among women with asthma, and then model that with environmental exposures. In that preliminary work, we did find that humidity was a potential influence that needs to be looked into further. Our team, with an engineering collaborative with the support of NSF, tested a novel smartphone compatible formaldehyde detection system using an existing passive sampling chromatography badge. We'll learn more about area sampling and methods in a later module, and how nurses tend to engage in that science. Nurses frequently innovate civic engagement and community-based participatory research methods, citizen science efforts, providing methodology support, strengthening citizen science methods, and creating sort of a structure of credibility and discerning research questions that are community-driven, integrating symptom science and continuous health monitoring and data use, and communicating with decision makers. This can include the use of symptom diaries, which nurses are expert at. If you work in this area, you might want to identify the sequence. Was this done first, second, or third, to again consider where there's potential to connect with new collaborators, what citations or literature you need to dive into, what was the time frame for this for you, or what research products came out, and what was the context. So again, note those light blue ovals of application and synthesis ring that we just discussed. These are very common in nursing and clinical science as phase 1 trials, systematic review, novel tool measurement, test, development. Go ahead and circle the notes that relate to your own research trajectory or plan. All right, we're on to the third ring of implementation and adjustment. This is where we do rigorous hypothesis testing. In real-world settings, adjust the products. Is it a new intervention? Is it a new tool? Is it a new measurement tool that we need to validate? A new method, a new treatment? You really want to account here for differences in settings and populations, elucidating, is this a tool that contributes to health disparities or helps to reduce them and eliminate them? It may be a new biomarker screen or assay validation, right? So in the last step, we talked about development. Now we're talking about validation. Clinical testing, effectiveness testing. What's important about the TRF, in contrast to how we commonly think about translational trajectories, is if you're testing dissemination interventions or implementation interventions, it fits here as hypothesis testing. If it's a new intervention, so for an example might be you're working on decarbonizing the health sector, and you're taking the best evidence and you're doing a workshop on that, and you're testing the effectiveness of that workshop and disseminating knowledge and practice change. So that's something new that you're testing, as opposed to taking an existing guideline or framework and implementing that into practice, which we'll get to next. So again, a difference between implementation science testing and applying to practice, which is evidence-based practice. So again, just like before, consider if you've already done work in this space, collaborators, citations, time frame, and context. And here you'll see on the screen are the nodes. And we circle back to the full model, and you can see where it fits in the entire translational continuum, and circle the work that relates to both what you have done and link to your research products, your publications, your presentations, your grants, and or circle the stage that you want to go to next. Of course, practice is moving evidence into common practice. This is not testing implementation, but the actual practice of implementation and sustained change. Again, I just want to clarify, in implementation science, we're testing strategies or interventions to translate to practice. This is the actual practice change. This may be using evidence from new guidelines, formalizing or codifying new public health interventions, instituting local, regional, state, or national policy practices, or motivating behavior change based on best evidence at the individual, family, group, or population levels. Clinical nurses who use evidence-based practice live in this space. Patient education, public health education, motivating patient behavior change are all examples. We're not testing these, we're actually doing them. This is the action step. One of the examples that we published in Nursing Outlook was my team authored a climate change resolution for one of my nursing organizations, the Emergency Nursing Association. If you have work in this area, again, you want to identify, was this done first, second, or third? Who were your collaborators? Link to the citations and projects. Think about the time frame and your context of support, because you may need to replicate where you've been in a previous setting, or pull in additional supports, or consider what made that successful. So again, in the dark blue ring of practice, you'll see the 6 circles of the nodes that we just discussed. Go ahead and circle and apply those to the products of your trajectory or your plans. Last but not least, we want to talk about broader impact. This is a change in outcome or obtaining those intended results of practice, which we know doesn't always happen. So Dr. Amiri in our Nursing Outlook publication talks about her community-based participatory research project to reduce exposure to lead and hookworm in Alabama. In my work, there's an example of eliminating new exposures from a criminally polluting factory. We've also done some work on looking at potential gas leaks from gas pipelines and reducing that potential exposure. This may be facility or population improvements in something like asthma outcomes. It may be a clinical impact as well. If you have work in this area, again, as part of applying it, you're going to consider what sequence this was done in your trajectory, what are your collaborators, what are those products you can tie it to, your citations and publications, your grants, your curriculum change, and what was the context or support you needed to succeed. Again, here's the whole model. Those 4 triangles on the outside, we've now walked through all of the rings and nodes, and then how you would tie your own work to this to create your own cartography. So translation, according to Pettibone et al. In 2018, is movement from one ring to another or between nodes on a ring, and a bridge is when 2 completed projects cross rings or nodes. So these are some things to consider as we translate or we speed the translation of research to impact, or fundamental questions to impact, and all of the community roles that researchers and educators have along that way. And these are questions we might want to ask in that planning phase of how do we maximize the impact of our work. So an example might be that a nurse scientist is looking at the NIEHS data set, which is a population level publicly available data set, and notes higher levels of biomarkers of exposure within a historically marginalized racial or ethnic group, and that may lead to some discovery or looking for a novel questionnaire screening tool to find out more, or a novel assay. In our current work, we took a secondary data analysis of racial disparities in a FEMA data set of household disaster preparedness which again is fundamental question, and applying that findings and looking deeper in a systematic review of interventions. So many examples of how we create bridges and link a project further towards impact. Here's an example from our published work. This is Dr. Huntington-Moskos example, and this was a grant, in planning a grant application, and I just want to emphasize how useful this can be, particularly in the R01 grant writing phase, when you have to link and talk about how your previous work and your previous successes link to the current project. So very useful in environmental health clinical sciences for that purpose. Place in your grant, place in how you're justifying how you're qualified and how this work is on a continuum. This was an example from several projects in my research. In this case, I want to just point out that the sequential depiction didn't work well, the what came first, what came second, because there were many projects started long before and ended long after others, and there was a lot of cross-pollinating across disparate areas, which all really led to some impact. But just to point out, in translating that and individualizing the TRF to my work on multi-level measurement and reduction of airborne pollutants with emergency effects in respiratory and sense organs, I had to modify the process a bit. And then here's Dr. Azita Amiri's example of a project. So, we talked about a grant application, a plan project, a career trajectory, and completed work. And then here we're looking at one community-based participatory research project that was completed. Again, more detailed explanations, please reach out to the workshop faculty and planners if you have questions about their particular trajectories, but I want you to have these examples. So don't forget step 4. Step 4 is, how is this helpful as a shared mental model? It really does a beautiful job of integrating a complexity, right? Methods and problems outside of traditional clinical sciences and in the medication and medical device development trajectory, and resonates with environmental exposure work and human health. It does have this complex nonlinear feeling to it, which can be very pragmatic for those who have those kinds of trajectories. And you can skip rings or nodes. There are note your areas of confusion or struggle where you had to iterate for model clarity. For example, for me, I've done a lot of program evaluation work, and those designs are outside of controlled trials, right? Is that a fundamental question? Should that be an application and synthesis? Is it practice because it's an evaluation of practice? I'm still grappling about where that best fit. It may come down to the program evaluation design, but it may be an opportunity for continued clarity as well. I also found that novel tool development with nurse engineering partnerships can be a great place to put in that fundamental question category and give it more clarity and life. And so, like any useful model, there's going to be iterations of it and improvements of this theory over time, and please consider being a part of it. We want to take your finished career cartography and send it into Dr. Pettibone's team for their consideration of how it's used and what improvements might be made. Your outcome during our workshop time together is to generate and submit your own TRF career cartography figure by the end of the workshop. You'll be submitting it through the virtual classroom, and we'll work on it as a team in the workshop. Here are ways to prepare. There are 2 worksheets in the virtual classroom to use for reflection, preparation, and dialogue. These can also be found at the NIEHS website link in the virtual classroom. Again, we're going to share this cohort's completed figures with the TRF creator, that lead author, Dr. Pettibone. So please include your name and contact information on the slide or an accompanying slide. We'll workshop the cohort's cartography together and ask that you submit during or immediately after the workshop to the online classroom. Just as a pragmatic note, you can pull a PDF of the whole TRF from the NIEHS website. If you're not able to edit PDFs, we did have to recreate the figures in PowerPoint and actually create our own rings and nodes in the work that we had done. So we'll put some starters as PowerPoint figures there for you, but most likely you'll be working through your PowerPoint slide. So with that, please bring your questions to the workshop. Please include them in the Google Classroom. There are no right or wrong answers. We're creating a community of practice here where we learn and improve from one another and help to clarify one another's ideas. I am so glad you're here and look forward to interacting as we develop your cartography figure. Thank you.